Monocotyledons, commonly referred to as monocots, are flowering plants whose seeds typically contain only one embryonic leaf, or cotyledon. They constitute one of the major groups into which the flowering plants have traditionally been divided. The rest of the flowering plants having two cotyledons and therefore classified as dicotyledons or dicots. However, molecular phylogenetic research has shown that while the monocots form a monophyletic group, a clade, the dicots do not. Monocots have almost always been recognized as a group, but with various taxonomic ranks and under several different names. The APG3 system of 2009 recognizes a clade called monocots, but does not assign it to a taxonomic rank. The monocots include about 60,000 species. The largest family in this group by number of species are the orchids, with more than 20,000 species. About half as many species belong to the true grasses, who are economically the most important family of monocots. In agriculture, the majority of the biomass produced comes from monocots. These include not only major grains, but also forage grasses, sugar cane, and bamboos. Other economically important monocot crops include various palms, bananas, gingers and the relatives turmeric and cardamom, asparagus and the onions and garlic family. Additionally, most of the horticultural bulbs, plants cultivated for the blooms, are monocots, such as lilies, daffodils, irises, amaryllis, cannas, bluebells and tulips. Description: the monocots or monocotyledons have a single cotyledon, or embryonic leaf, in the seeds. Historically, this feature was used to contrast the monocots with the dicotyledons or dicots, which typically have two cotyledons. However, modern research has shown that the dicots are not a natural group. From a diagnostic point of view, the number of cotyledons is neither a particularly useful characteristic, nor is it completely reliable. Comparison with dicotyledons: the traditionally listed differences between monocotyledons and dicotyledons are as follows. This is a broad sketch, only not invariably applicable, as there are a number of exceptions. The differences indicated are more true for monocots versus eudicots. A number of these differences are not unique to the monocots, and while still useful, no one single feature will infallibly identify a plant as a monocot. For example, trimerous flowers and monosulcate pollen are also found in magnoliids, of which exclusively adventitious roots are found in some of the Paperaceae. Similarly, at least one of these traits, parallel leaf veins, is far from universal among the monocots. Monocots with broad leaves and reticulate leaf veins, typical of dicots, are found in a wide variety of monocot families. For example, Trillium, Smilax, and Pogonia, and the Dioscoreales. Potamogton are one of several monocots with tetramerous flowers. Other plants exhibit a mixture of characteristics. Nymphaceae have reticulate veins, have single cotyledon, adventitious roots and a monocot-like vascular bundle. These examples reflect the shared ancestry. Nevertheless, this list of traits is a generally valid set of contrasts, especially when contrasting monocots with eudicots rather than non-monocot flowering plants in general. The vascular system: monocots have a distinctive arrangement of vascular tissue known as an attictostula, in which the vascular tissue is scattered rather than arranged in concentric rings. Collenchyma is absent in monocot stems, roots and leaves. Many monocots are herbaceous and do not have the ability to increase the width of a stem via the same kind of vascular cambium found in non-monocot, woody plants. However, some monocots do have secondary growth, and because it does not arise from a single vascular cambium producing xylem inwards and phloem outwards, it is termed anomalous secondary secondary growth. Examples of large monocots which either exhibit secondary growth or can reach large sizes without it are palms, screwpines, bananas, yucca, aloe, dracaena, and cordyline. Synapomorphies, by contrast, Douglas E. Soltis and others identify 13 synapomorphies: calcium oxalate rhaphides, absence of vessels in leaves, monocotyledonous anther wall formation, successive microsporogenesis, syncarpus gynoecium, parietal placentation, monocotyledonous seedling, persistent radical, haustorial cotyledon tip, opened cotyledon sheath, steroidal saponins, fly pollination, diffuse vascular bundles, and absence of secondary growth. Taxonomy. The monocots form one of 5 major lineages of mesangiosperms, which in themselves form 99.95% of all angiosperms. The monocots and the eudicots are the largest and most diversified angiosperm radiations, accounting for 20% and 75% of all angiosperm species respectively. Monocot diversity includes perennial geophytes including ornamental flowers, rosette and succulent epiphytes, mycoheterotrophs, all in the lilioid monocots, major grains in the grass family, as well as woody tree like palm trees and bamboo in the cotyledon monocots, as well as both emergent and floating or submerged aquatic plants. Early history. The monocots are one of the major divisions of the flowering plants or angiosperms. They have been recognized as a natural group since John Ray's studies of seed structure in the 17th century. Ray was the first botanical systematist, and in his examination of seeds, first observed the dichotomy of cotyledon structure. He reported his findings in a paper read to the Royal Society on the 17th of December 1674, entitled A Discourse on the Seeds of Plants. Since this paper appeared a year before the publication of Malpighi's Anatomy Plantarum, Ray has the priority. At the time, Ray did not fully realize the importance of his discovery but progressively developed this over successive publications, and since these were in Latin, seed leaves became folia samonalia and then cotyledon. Following Malpighi, Malpighi and Ray were familiar with each other's work, and Malpighi, in describing the same structures, had introduced the term cotyledon, which Ray adopted in his subsequent writing. In this experiment, Malpighi also showed that the cotyledons were critical to the development of the plant, proof that Ray required for his theory. In his Methodus Plantarum Nova, Ray also developed and justified the natural or pre-evolutionary approach to classification, based on characteristics selected a posteriori in order to group together taxa that have the greatest number of shared characteristics. This approach, also referred to as polythetic, would last till evolutionary theory enabled Euclid to develop the phyletic system that superseded it in the late 19th century, based on an understanding of the acquisition of characteristics. He also made the crucial observation xhac salmonum divisiona summum potus generalis plantarum distinctio, hic meo judicio omnium primare lunge optima in easi. Que plantula seminale sunt bifoliae ut delta iota lambda omicron beta omega egwe plantula sem. Adulta analoga, that is, between monocots and dicots. He illustrated this by quoting from Malpighi and including reproductions of Malpighi's drawings of cotyledons. Initially, Ray did not develop a classification of flowering plants based on a division by the number of cotyledons, but developed his ideas over successive publications, coining the terms monocotyledones and dicotyledones in 17 1903, in the revised version of his Methodus, as a primary method for dividing them, Herber floriferae, dividipos and addiximus, in Monocotyledones and amp. Dicotyledons. Although Linnaeus did not utilize Ray's discovery, basing his own classification solely on floral reproductive morphology, every taxonomist since then, starting with de Jussi and de Candolle, has used Ray's distinction as a major classification characteristic. Modern era. Modern research based on DNA has confirmed the status of the monocots as a monophyletic group, a clade. In contrast to the other historical divisions of the flowering plants, which have had to be substantially reorganized, the monocots form about a quarter of all of the angiosperms. Of some 60,000 species, by far the largest number are found in two families, the orchids and grasses. The orchids contain about 25,000 species and the grasses about 11,000. Other well-known groups within the Poales order include the Cyperaceae and Juncaceae, and the monocots also include familiar families such as the palms and lilies. Taxonomists had considerable latitude in naming this group, as the monocots are a group above the rank of family. Article 16 of the ICBN allows either a descriptive name or a name formed from the name of an included family. Historically, the Monocotyledons were named Monocotyledonii in the de Candolle system and the Engler system, Monocotyledones in the Bentham and amp. Hooker system and the Wettstein system, class Liliopsida in the Taxonomy system and the Cronquist system, subclass Liliidae in the Dahlgren system and the Thorne system, clade monocots in the angiosperm phylogeny group systems, the APG system, the APG2 system, the APG3 system and the APG-IV system. Until the rise of the phylogenetic APG systems, it was widely accepted that angiosperms were neatly split between monocots and dicots, a state reflected in virtually all the systems. It is now understood that various groups, notably the magnoliids and ancient lineages known as the basal angiosperms, fall outside of this dichotomy. Each of these systems uses its own internal taxonomy for the group. The Monocotyledons are famous as a group that is extremely stable in its outer borders, while in its internal taxonomy is extremely unstable. Molecular studies have both confirmed the monophyly of the monocots and helped elucidate relationships within this group. The APG3 system does not assign the monocots to a taxonomic rank, instead recognizing a monocots clade. However, there has remained some uncertainty regarding the exact relationships between the major lineages, with a number of competing models. Subdivisions. Historically, Bentham considered the monocots to consist of four alliances: epigyno, coronarii, nudiflorae and glumales, based on floral characteristics. He describes the attempts to subdivide the group since the days of Lindley as largely unsuccessful. Like most subsequent classification systems, it failed to distinguish between two major orders, Liliales and Asparagales, now recognized as quite separate. A major advance in this respect was the work of Rolf Dahlgren, which would form the basis of the angiosperm phylogeny group's subsequent modern classification of monocot families. Dahlgren, who used the alternate name Liliidae, considered the monocots as a subclass of angiosperms characterized by a single cotyledon and the presence of triangular protein bodies in the sieve tube plastids. He divided the monocots into 7 superorders: Alasmatoflorae, Araflorae, Triuridiflorae, Liliiflorae, Zingibiriflorae, Camelliniflorae and Araciflorae. With respect to the specific issue regarding Liliales and Asparagales, Dahlgren followed Huber in adopting a splitter approach, in contrast to the long-standing tendency to view Liliaceae as a very broad sensu lato family. Following Dahlgren's untimely death in 1987, his work was continued by his widow, Gertrude Dahlgren, who published a revised version of the classification in 1989. In this scheme, the suffix flori was replaced with an e and the number of superorders expanded to 10 with the addition of Bromeliani, Cyclanthini and Pandarn. The APG system establishes 10 orders of monocots and 2 families of monocots not yet assigned to any order. More recently, the Petrosaviaceae has been included in the Petrosaviales and placed near the lilioid orders. The family Hydatilaceae, assigned to order Poales in the APG2 system, has since been recognized as being misplaced in the monocots, and instead proves to be most closely related to the water lilies. Family Nymphaceae. Family Dasypogonaceae is placed in order Echales in the APG-IV system. Evolution. The monocots form a monophyletic group arising early in the history of the flowering plants, but the fossil record is meager. The earliest fossils presumed to be monocot remains date from the early Cretaceous period. For a very long time, fossils of palm trees were believed to be the oldest monocots, first appearing 90 million years ago, but this estimate may not be entirely true. At least some putative monocot fossils have been found in strata as old as the eudicots. The oldest fossils that are unequivocally monocots are pollen from the late Barremian-Aptian, early Cretaceous period, about 120–110 million years ago, and are assignable to clade Poddoideae monsterii Araceae, being Araceae, sister to other Alismatales. They have also found flower fossils of Trieridaceae in upper Cretaceous rocks in New Jersey, becoming the oldest known sighting of saprophytic mycotrophic habits in angiosperm plants and among the oldest known fossils of Monocotyledons. Topology of the angiosperm phylogenetic tree could infer that the monocots would be among the oldest lineages of angiosperms, which would support the theory that they are just as old as the eudicots. The pollen of the eudicots dates back 125 million years, so the lineage of monocots should be that old too. Molecular clock estimates. Carl Bremer, using RBCL sequences and the mean path length method, estimated the age of the monocot crown group as 134 million years. Similarly, Wickstrom et al., using Sanderson's non-parametric rate smoothing approach, obtained ages of 158 to 141 million years for the crown group of monocots. All these estimates have large error ranges, and Wickstrom et al. Used only a single calibration point, namely the split between Fagales and Cucurbitales, which was set to 84 Mart in the late Santonian period. Early molecular clock studies using strict clock models had estimated the monocot crown age to 200 plus or minus 20 million years ago, or 160 plus or minus 16 million years, while studies using relaxed clocks have obtained 135 minus 131 million years, or 133.8 to 124 million years. Bremer's estimate of 134 million years has been used as a secondary calibration point in other analyses. Some estimates placed the emergence of the monocots as far back as 150 Maya in the Jurassic period. Core group. The age of the core group of so-called nuclear monocots or core monocots, which correspond to all orders except Dicorales and Alismatales, is about 131 million years to present, and crown group age is about 126 million years to the present. The subsequent branching in this part of the tree, including the crown Petrosaviaceae group, may be in the period around 125 65–120 million years BC, and stem groups of all other orders, including Cotyledons, would have diverged about shortly after 115 million years. These and many clades within these orders may have originated in southern Gondwana, i.e., Antarctica, Australasia, and southern South America. Aquatic monocots. The aquatic monocots of Alismatales have commonly been regarded as primitive. They have also been considered to have the most primitive foliage, which were cross-linked as Dioscoriales and Melanthelius. Keep in mind that the most primitive monocot is not necessarily the sister of everyone else. This is because the ancestral or primitive characters are inferred by means of the reconstruction of character states, with the help of the phylogenetic tree. So primitive characters of monocots may be present in some derived groups. On the other hand, the basal taxa may exhibit many morphological or topomorphies. So although Ecology is the sister group to the remaining Monocotyledons, the result does not imply that Ecology is the most primitive monocot in terms of its character states. In fact, Ecology is highly derived in many morphological characters, and that is precisely why Ecology and Alismatales occupied relatively derived positions in the trees produced by Chase al. And others. Some authors support the idea of an aquatic phase as the origin of monocots. The phylogenetic position of Alismatales, which occupy a relationship with the rest except the Ecology, do not rule out the idea, because it could be the most primitive monocots but not the most basal. The atictostular stem, the long and linear leaves, the absence of secondary growth roots in groups instead of a single root branching, including some podial use, are consistent with the water source. However, while monocots were sisters of the aquatic ceratophyllales, or their origin is related to the adoption of some form of aquatic habit, it would not help much to the understanding of how it evolved to develop the distinctive anatomical features. The monocot seems so different from the rest of angiosperms, and it's difficult to relate the morphology, anatomy and development and those of broad-leaved angiosperms. Other taxa. In the past, taxa which had petiolate leaves with reticulate venation were considered primitive within the monocots, because of its superficial resemblance to the leaves of dicotyledons. Recent work suggests that these taxa are sparse in the phylogenetic tree of monocots, such as fleshy-fruited taxa. The two features would be adapted to conditions that evolved together regardless. Among the taxa involved were Smilax, Trillium, Dioscorea, etc. A number of these plants are vines that tend to live in shaded habitats for at least part of their lives, and may also have a relationship with the shapeless stomata. Reticulate venation seems to have appeared at least 26 times in monocots, in fleshy fruits 21 times, and the two characteristics, though different, showed strong signs of a tendency to be good or bad in tandem, a phenomenon described as concerted convergence. Etymology. The name Monocotyledons is derived from the traditional botanical name Monocotyledons, which refers to the fact that most members of this group have one cotyledon, or embryonic leaf, in the seeds. Ecology Emergence. Some monocots, such as grasses, have hypogeal emergence where the mesocotyl elongates and pushes the coleoptile toward the soil surface. Since elongation occurs above the cotyledon, it is left in place in the soil where it was planted. Many dicots have epigeal emergence, in which the hypocotyl elongates and becomes arched in the soil. As the hypocotyl continues to elongate, it pulls the cotyledons upward, above the soil surface. Uses of the monocots. The grasses are of enormous economic importance as a source of animal and human food, and form the largest component of agricultural species in terms of biomass produced. Bibliography.